All right, everyone, we might just get started. So on behalf of my co-program manager, Quinn, and High Blood Pressure Council of Australia, we welcome everyone to day 2 of the Hypertension Winter School. Before we get started, I want to pay my respects to traditional custodians of the lands on which we meet today. I want to pay my respects to their elders, past, present, and emerging. So some housekeeping, please remain muted during presentations. There will be an opportunity to ask the speakers questions at the end of their presentations. Please also refer to page 3 of the program booklet for full Zoom etiquette. These presentations are being recorded. If you do not wish to be recorded, please turn off your video when asking a question. We also ask everyone who is not speaking today to turn off their videos to reduce bandwidth for everyone. If you would like to tweet about the event, ask that you use the official Twitter hashtag HBPRCA_WS2020, which will be posted in the chat. Today, we also have, and we're very excited about, a virtual networking session. There will be 2 6-minute slots for you to socialise and network with the other attendees. Attendees will be randomly assigned into breakout rooms, and we encourage all to participate in the virtual selfie competition. We also invite you to click on the link that's going to be posted in the chat shortly for more information about the networking event. I would now like to call on Quinn to chair our first session.
Thanks, Hamdi. So it's with great pleasure to introduce the first speaker for today, Professor Grant Drummond from La Trobe University. Grant, if you could share your screen.
No worries. If I just get my video, I can't get my video. Oh, there we go.
And so Grant, yeah, that's excellent. Just put on presenter mode.
Is that right? Is that working, Quinn? Or?
Looks fantastic. So Grant will be speaking about animal models of hypertension. So thank you, Grant.
No worries. Thanks, Quinn, and thanks to the organisers for giving me the opportunity to talk to you today. Before I get into this, I'd really just like to acknowledge the people that, well, probably that know a lot more about this topic than what I do, and who were very, very helpful in putting this presentation together. So there's 3 people in particular. Francine Marks from Monash University, who I'm sure you all know very well as an expert in experimental and clinical hypertension research. I saw Francine give a similar presentation earlier in the year, and she did a fantastic job of this. So I hope I can emulate that. And then from my lab, Dr. Anthony Vinn and Dr. Maria Jelenic, who are also very actively involved in actually doing the research in the lab, a lot more than what I am at the moment. So again, just thank you to these guys for helping me put together this talk today. Look, I guess, as you're all very, very well aware, hypertension is an extremely complex disease. A primary hypertension, an extremely complex disease. It's responsible for about 90 to 95% of cases of hypertension in general. This figure down here is from a beautiful article from Francine a few years ago that sort of, I guess, encapsulates the complexity that we're talking about here. Primary hypertension is a multifactorial disease. It involves numerous organs in the body: the kidneys, the heart, the vessels, the brain. You'll hear later from Bill that it involves the immune system, and Francine's done some beautiful work showing that the microbiome plays a great role. But of course, these systems also have interactions with the environment, and also the underlying genetics of the individual, meaning that we've got this really, really complex interplay of genetics, environment, and microbiome that are all contributing to the ultimate condition of hypertension. So I guess with that background in mind, it's not surprising then that there is no one model no one animal model can possibly replicate all of the aspects of primary hypertension. There are some very, very good models of secondary hypertension, particularly renal damage models, but no single model can represent all aspects of primary hypertension. And I guess it's for that reason that if we look through the literature, there are numerous models of hypertension reported on. I clearly can't talk about all of these today. And in fact, I certainly wouldn't profess to be an expert in all of these models either. So what I thought I would do is just perhaps focus on some of the more commonly used models of hypertension. I guess we could, just in general, hypertension models can be broadly divided into genetically based models and sort of induced models. That is, models of hypertension are induced either by changes to the diet, pharmacological interventions or surgical interventions. And in my talk today, I guess I'll cover just a sort of a smattering of across a few of these different models. Many of the models that I'll talk to you about today have been primarily performed in rodents, especially rats and mice. But you'll see that some of them could equally be applied to larger animal models, although not very commonly done so. So obviously, the reason that most of the studies are performed in small animals is mostly around practical things. They're cost effective, shorter lifespan, shorter gestation periods, high throughput, so you can just generate data a lot more quickly when you use smaller models. I guess a major advantage of smaller animals, rodents, especially mice, but more so now rats and rabbits, is the tractability of genetic manipulation. And also just the tools that are available for research. There's lots and lots of good antibodies available to mouse and rat proteins, so just easier things to work with. This is not to say that there isn't a need for larger animal models, obviously. Clearly, they're anatomically, physiologically, and hemodynamically more closely related to humans than small animals, and so perhaps more of the findings in larger animal models may be more applicable to the human state. I guess another practical consideration is that when you use mice, you really do struggle to get a lot of material to do your studies. So larger animals obviously mean that there's a lot more tissue or samples available for experimentation. Another problem in the hypertension field is, and probably not only the hypertension field, but a lot of fields, is that there's not very many good non-human primate models of hypertension. There's one monkey model that I know of, the Virat monkey, I think they're called, but that's certainly an area of need in this field. And I suppose when we talk about models of hypertension, we're not just talking about models that allow us to study blood pressure. Obviously, that's the primary endpoint that we tend to measure in our studies on hypertension. But our models of hypertension are also associated with a number of other of the known sort of complications of hypertension, including cardiac remodeling and dysfunction, renal injury and dysfunction, stroke, cognitive impairment, vascular remodeling and stiffening, vascular inflammation, endothelial dysfunction, and aortic aneurysm. Now, different models model the human condition, or these human conditions more effectively than others. So I guess when you're planning your studies, it's really, really important that you consider what is it the endpoint that you actually want to measure, and which model or models are the best for helping you or for studying that endpoint, the most valid for studying that endpoint. I think a really important thing to note is that we all know that one of the major outcomes of hypertension is the development of atherosclerosis and ultimately myocardial infarction. It's important to note that none of the hypertension models available are associated with the spontaneous development of atherosclerosis or acute myocardial infarction. To achieve that, you need to kind of superimpose your hypertension models onto other disease models, such as the apolipoprotein knockout mice. So that's also been a little bit of a drawback in the field. All right, so as I said, I'm just going to run you through a few of the different models in the next sort of 10 minutes. I'll probably focus mainly on the ones that I'm most familiar with, and then at the end, perhaps summarise where I think we're at and what we might need to think about going forward. So by far and away, at least over the last decade or so, the most commonly used model of hypertension is this pharmacological model involving angiotensin II infusion, mostly into mice, but equally it works in rats as well. I'm sure I don't need to talk to this audience about what we're trying to achieve when we do this. You all know that angiotensin 2 is the main effector hormone of the angiotensin, the renin-angiotensin pathway, and it acts primarily on AT1 receptors to mediate a host of effects that ultimately result in hypertension. Aldosterone secretion, ADH secretion, vasoconstriction. Angiotensin is a very powerful vasoconstriction, and it also increases sympathetic activity. So I guess when we're infusing angiotensin 2 into animals, we're trying to sort of replicate a number of these physiological changes that are known to occur in hypertension. Something that is probably worth noting is that angiotensin 2 actually has multiple receptors that it acts on, more than just the two, but probably the most well-described receptor other than the AT1 receptor is the AT2 receptor, which is via various mechanisms counteracts a lot of the effects of the AT1 receptor. And in the next slide, you'll see why that's relevant. So some of the features of the angiotensin 2 models are that obviously mice are chronically treated with angiotensin 2 via implantation of one of these subcutaneous osmotic mini pumps. So you load the mini pump up with a certain concentration of angiotensin 2, surgically implanted into the mouse, and then you can maintain that infusion anything up to 8 weeks. It induces moderate to severe renin-angiotensin aldosterone system-dependent hypertension. And following between 2 and 4 weeks of treatment, you start seeing evidence of end organ damage, including cardiac and vascular remodeling, vascular dysfunction and inflammation, and you see some renal changes, but they are very, very mild in this model. The control mice for these studies are mice that receive an osmotic mini pump, similarly to the ANG mice, but instead filled with the vehicle for angiotensin 2. Some of the advantages of the model: it's the most widely used preclinical model of hypertension currently, as I said earlier. The severity of the disease can be manipulated via different doses and treatment durations. Up here in this graph, you can see that you do see a nice dose-dependent effect on blood pressure and systolic blood pressure with increasing doses of angiotensin 2. You do ultimately reach a ceiling effect when you with angiotensin 2. This dose down here, this low dose of angiotensin 2, around about the 0.2 milligrams per kilogram per day, is often referred to as a sub-pressor dose or a slow pressor model. The advantage of this model, perhaps over the other models, is that you get this period of kind of a pre-hypertensive state, which is kind of a window of opportunity to look to see what might happen, what some of the changes that might be occurring before blood pressure starts going up. I think another interesting observation in this model is that you see quite different effects in males versus females. And this is studies out of Kate Denton's lab, by Russell Brown, I think, performed the studies, showing that male mice have a much more severe hypertensive response than do female mice. And this appears to be due to these opposing effects of the AT2 receptor, which are more prominent in female mice. When you knock out the AT2 receptor from female mice, you can actually enhance the hypertensive response. And I guess at this point, I'd just comment that a major limitation in our field, and along with a lot of fields of research in the cardiovascular area at least, is the preponderance of preclinical work on male subjects. I think a lot of us, and indeed our lab's been guilty of this in the past, have sort of just assumed if you study male mice, you'll get all the information you need to hopefully translate your findings down the track. But that's obviously not the case, and there are major differences. And we really do need almost a paradigm shift, I think, in the way we approach this and make sure that we're always studying males and females in our studies. I'll move on now. So this is another pharmacological model that's not as commonly used, but nonetheless, due to its convenience or ease of establishing is quite a useful model, and that involves the administration of L-nitroarginine methyl ester into mice. Now, L-name, for those of you who are not familiar with the pathway, is an inhibitor it's an analogue of L-arginine, and thus blocks nitric oxide synthase activity. And what we're trying to achieve with this model is to block the production of endothelial nitric oxide, which produces vasorelaxation, as you all know, and thus by doing that we can presumably increase peripheral vascular resistance and thereby increase blood pressure. And you can see that it does have a nice robust effect on blood pressure. As I said, some of the advantages of this model is it's minimally invasive. LMAIM can be administered very, very simply via the drinking water. But a limitation of the model is that, that there are other nitric oxide synthase enzymes in addition to eNOS, including inducible nitric oxide synthase. You may be aware that inducible nitric oxide synthase is an inflammatory molecule that produces large amounts of nitric oxide, which actually contribute to oxidative stress. And so in some models, this infusion of LMAIM can actually afford protection against certain organ damage. A way around this, and I'll mention this just a little bit later in the talk, is that is to look at eNOS knockout mice rather than using a pharmacological approach to specifically look at what happens to blood pressure and organ damage when you target the gene endothelial nitric oxide synthase. Another nice pharmacological model that provides a point of difference from the angiotensin model are these mineralocorticoid salt models of hypertension. In these models, what we're really trying to achieve is stimulation of the mineralocorticoid receptor in the distal collecting tubules and collecting ducts, which in turn upregulates sodium reabsorption mechanisms in those cells and thereby promotes volume overload. It involves union nephrectomy of mice and rats, and then infusion of a mineralocorticoid, whether that be deoxycorticosterone acetate in this example. You can also use aldosterone. The other aspect of the model is it's a salt-sensitive model, so it involves increasing the salt intake of mice by adding it to the drinking water. Induces moderate to severe hypertension with quite profound end organ damage, including renal, cardiac, and vascular damage. And some of the advantages of the model, as I said before, it's a volume overload model with low circulating renin. So when performing studies in the angiotensin model, it provides a nice point of sort of contrast to that model in that you can sort of make conclusions about whether your observation is an angiotensin-dependent effect versus a sort of a more generally applicable hypertension effect. The limitations of the model, it is quite technically demanding, the union nephrectomy surgery. It takes quite a lot of practice to get that right and thus to get reproducible outcomes. And the other problem with the model is that renal function is a little bit difficult to interpret. The mice drink excessive amounts of saline, and therefore, as they become hypertensive, they actually excrete a lot larger volume of urine and electrolytes. So not what you would often see in a hypertension model, where you might expect it to be sort of retention of sodium and volume. A little thing here, I guess we've just recently started analysing the model in both males and females, and whilst there's some evidence in rats that rats may be more susceptible to the hypertensive effects of doka salt, it seems in mice that we see similar sort of sensitivity to the treatment in both males and females. Just very quickly, Maria Jelenic is an expert in this model, which involves feeding animals a high-fat diet for generally for greater than 10 weeks. And when you do this, mice and rats and rabbits will develop spontaneous hypertension. It's not a very profound model of hypertension. They only develop about a 10 millimetre increase in blood pressure, but nonetheless, it's quite a reproducible response. The other advantages are that the mice develop other comorbidities that are often associated with hypertension, high BMI, dyslipidemia, hyperglycemia. It's also technically quite a simple model as well. The limitations, I guess one of the limitations is that the blood pressure change is not very dramatic, which means it can be difficult to detect the blood pressure change with tail cuff studies. Often you need to employ more expensive radiotelemetry to see robust or measure robust changes in blood pressure. There are strain and species differences in the resistance or susceptibility to developing hypertension. And I think probably another drawback of these sorts of models is they do take a long time to develop. So to develop hypertension, so it's quite slow to generate data. I can see I'm running out of time here, so I'll just get a bit of a wriggle on here. So the genetic models, the ones I just spoke to you, are sort of a combination of pharmacological models and surgical models, I guess. Genetic models or polygenetic models, most of these have been developed in rats, although there is a nice model I'll mention in a second in mice. And probably the most widely studied, and the one you're all aware of, is the spontaneously hypertensive rat. Now, these mice originated from the cross of an outbred Worcester male rat that was found to have spontaneously elevated blood pressure with a female that had slightly elevated blood pressure. Subsequent brother-sister crosses on animals with systolic blood pressure greater than 150 millimetres of mercury were ultimately used to develop this strain. Hypertension begins to develop around 5 to 6 weeks of age and reaches systolic pressures of quite high, of around 120 to 200 millimetres of mercury. And there are also some substrains available in this model, such as the stroke-prone SHR. The advantages of the model, I guess similar to human hypertension, it's a polygenetic model or a polygenic model. It's often considered the gold standard rodent model for essential hypertension. It's reliable and it produces a very reproducible phenotype in terms of blood pressure, hypertrophy, renal injury, cardiac hypertrophy and renal injury. And another, I guess another indication that the model is of value is that all known antihypertensive drugs lower blood pressure in the SHR. The limitations of the model, behavioural studies are difficult as these rats are actually hyperactive. They're also used as a model of ADHD actually. Whilst it's a polygenic model like human hypertension, the genetics are quite different from humans obviously. Most of the loci are homozygous, which is very unlike humans. And perhaps the biggest drawback of these, and indeed all polygenic models of hypertension, that you really don't have an appropriate control strain. If you think about it, these mice were bred over 60 years ago, so there's been 60 years of genetic drift from the control Worcester Kyoto strain. So they're really no longer a good genetic control for SHRs. And this is evident not in terms of differences in non-hypertension related phenotypes, such as growth rates between the two animals, the two strains. I'm not going to go, in the interest of time, I'm not going to go into this model. This is another polygenic model of hypertension developed by Lewis Dahl, in which he selected for mice that showed a hypertensive response to high salt diet, the Dahl salt sensitive rat. Like the SHR, the major limitation of this model is the lack of an appropriate control strain, given the great deal of genetic drift that's occurred over the last, for this model, about 50 years. There's other people in the audience that certainly have more expertise with this model than me, but this is the BPH model. This is a mouse model of spontaneous hypertension. Again, these mice were selected on the basis of demonstrating higher blood pressure. So it was selective breeding of mice that had higher blood pressure to produce the hypertensive strain. Again, in the interest of time, I won't go too deeply into this, but the limitations, the main limitation again of the model is the difficulty in controlling or finding a suitable control given the genetic drift that's occurred. I think I'll just skip through this. I really just want to get to these last couple of slides now. And I think one of the other really important things that we have to consider with our animal models of hypertension is the way that we measure blood pressure. This is really one of the great bones of contention whenever we publish a paper, try to get a paper published, we get criticised for the way we measure blood pressure. Now, there are probably two broad ways of measuring blood pressure in animal models. The first is the direct measurement of blood pressure involving sort of intra-arterial measurement of blood pressure. And the gold standard way of doing this is through radiotelemetry, in which a blood pressure probe is inserted either into the abdominal aorta or into the carotid arteries. The animals are sewed up or stitched up again and allowed to then freely move around their cage on a receiver, such that you can measure blood pressure continuously over a period for up to sort of 8 to 10 weeks. We've done this. The advantage that this model has is that it allows you to measure all sort of aspects of blood pressure: systolic blood pressure, diastolic blood pressure, mean arterial pressure, diurnal blood pressure patterns. But the disadvantage of the technique is that it's highly invasive. It requires post-surgical antibiotics and analgesia, which can obviously influence your experimental findings. And it's a very, very difficult technique to master, and I'm sure anyone who does telemetry will attest to it's extremely expensive. Contrast that technique, I guess, with the more routinely used, I'm nearly finished now, Quinn, the more routinely used tail cuff blood pressure method. This is a non-invasive way of measuring blood pressure in conscious but restrained animals. It can be used in both long and short-term studies for repeated measurements. It's high throughput. You can do multiple animals at the same time. It requires little specialised training. It's very cost effective. But obviously restraint is stressful on mice. And although this can be to some extent ameliorated through acclimation and training of the animals and also the operators to make sure that they don't stress the mice out too much. It's more variable than the direct measures of blood pressure. And also, I think a major issue with this is it can be subject to bias. So it's very important that blinding's adopted in the experiments using this model. I guess an important aspect of this, though, it is very analogous to how we measure blood pressure in humans most commonly through an arm cuff. So, all right. So I guess just to conclude now, primary hypertension remains a really difficult disease to treat, as I'm sure all of us are very well aware with current drugs. Blood pressure remains high in approximately 50% of treated patients, and in maybe 10 to 15% of those patients, they truly have resistant hypertension. And even when we do lower blood pressure in patients, there is an element of residual risk in those patients still, cardiovascular risk. I guess a little bit of an indictment on our field is that there's no new class of hypertensive drugs that entered the market since the 1990s, despite the fact that we're still not treating this condition well. And I think some of this is possibly around the limitations of the models of hypertension we have. As I said, most models only mimic one or a few aspects of primary hypertension. No model really reflects resistant hypertension. No models are associated with the spontaneous development of atherosclerosis or acute myocardial infarction, and there are very few validated multifactorial models of essential hypertension in non-human primates. That's one issue, but I think it doesn't matter how good your models are, unless you employ good experimental design practices, such as the appropriate choice of models, animal ages, male and females being included in experiments, blinding, randomisation. I think this is also another reason that we've struggled a little to translate in the field, and all labs, including our own, have probably been guilty in the past of not adhering to the best experimental design protocols. So we need to get better at that. I know I've gone over time. I'm really sorry about that. I'll leave it at that, and thank you all for listening.
Thanks so much, Grant. That was a really fantastic and really detailed presentation, and I'm sure everyone learned a lot. We have time for maybe a couple quick questions. So I noticed someone has a raised hand. I think that's Rivati, if you would like to ask your question.
Hi, Grant. Thanks for your very concise and nice presentation. Which animal model would you suggest that could closely, a small animal model, I understand the larger the better, could closely mimic human hypertension scenario, especially to study vascular remodeling and other things?
Look, I think probably the take-home message there is that no one model will replicate all aspects of human hypertension. The approach that we take most of the models are associated with vascular dysfunction, vascular remodeling and vascular dysfunction. The approach that we take in our lab is that we use the angiotensin model kind of as our go-to model. And when we make interesting observations in that model, we try to validate them with another model. And as I sort of mentioned, the DOCA salt model provides a kind of nice contrast to being a low renin model. A salt-sensitive model provides a nice contrast to the angiotensin model. So that's the approach that we take in our lab. We kind of try to validate key findings from the Ang2 model in DOCA salt. But like I said, there's no one model that really replicates all elements of primary hypertension.
Great, thank you. And one last question from Matt Snelson. How do genetic models of obesity, example Zucker rat or the OB mice, affect hypertension, and can these be used as models?
Look, I don't have a lot of experience with the DBDB or OBOB mice. My understanding is that blood pressure doesn't change very much in those models, but there might be somebody else in the audience that's more qualified to answer that than me. But if it does, it's a very, very mild effect.